I do want to very much thank the people who have helped fund research over the years. The FDA has helped us, and especially Cynthia has been a tremendous project manager, and she's going to talk about the goals of the EPA and the work that was done here. I forgot to put in a slide thanking all our sponsors. We hope to have more, okay? Right now at the NIH, I think it's significant. I'm going to get to this in just a minute. By the way, this is our team. I think a lot of you probably know Suzanne Tuber, who's an expert in the clinical side of nut reactivity, and supplied a lot of the serum we're going to talk about. So Hila's sitting here, so, yeah. And so if the picture, she verified this picture, and of course, so video, and then my good friend, Dr. Terumi Midoro Horiyuti, who actually got me into this field in the first place. I'm actually more a virologist, and I keep sliding back into virology because a lot of the things that we're looking at with allergens also have applications in vaccine design. So what I'm going to tell you about, and this will be a much shorter talk, of course, because I don't have to cover, Dr. Brown took care of the equation side of things, as you saw, and the charts and things. So what I'm going to be telling you about is simply the practical applications. And what I've been interested in is how proteins that are quite different in their primary sequence can actually cause cross-reactions, and can we identify discrete areas of these proteins that would be specific for these cross-reactions. And also, are there properties of peanuts and nuts that make them special? And I'm going to show you some experimental verification of the PD scale that Dr. Brown explained to you. So I think one of the big problems in allergy research has been that up until about the 1970s, allergy was considered a female neurosis rather than an actual disease. And I'm basing this on I belong to the Osler Club at the UTMB. Those of you may know that William Osler, back around 1890, published what is still considered the first medical textbook. And Osler dedicates so diabetes actually is very well described in Osler's book at that time. But allergy, not so well. He gives 4 pages to something he calls at that time hay, or parentheses rose fever. And he says that the first thing that he has noted, that people with allergy tend to be female, tend to be somewhat neurotic. He uses the term hysterical, which was acceptable at that time. And the basis for his evidence is an experiment that was done sometime in the 1880s, where they took a poor woman who was complaining of rose fever and sneezed whenever she came near a rose, and they gave her an artificial rose to sniff, and she sneezed. And they said, therefore, of course, it was all in her head. Now, first of all, think about this experiment. The rose that she was given was probably made in a floral shop. All right, so that's one of the things. But the second thing was that if in the 19th century was crazy about rose water. All of the Oriental sweets had rose water in them. Rose and rose creams, and rose was everywhere. And it is quite likely that this person designing this artificial rose would have dipped it in rose water. So the experiment to begin with was flawed. But it had horrendous consequences. And it really wasn't until the 1970s or so that allergy was considered to be a respectable area to study. Now, from that time on, all of a sudden, there were cases acknowledged of people dying of exposure to allergens, particularly peanuts. And from around the 1990s, there was really a concerted effort in a few groups throughout the world to find proteins and to actually establish a chemical basis for allergy. And of course, an immunological basis as we got better with immunology. And so I think our tools, what we're doing now is the role of computers in allergy. It had to come eventually, it's just a bit later. So one of the things that's very intriguing, of course, is for a long time, people were talking about multiple allergy syndrome. And again, the emphasis was always on females and neurosis when it came up, when really it probably wasn't, because the same proteins, these same allergens, can turn up in many different forms. Now, I told you that Terumi got me into the allergy field. It was that they were having difficulty at that time isolating JUNA1. It was one of those intractable proteins. And of course, JUNA1 was this great big peak, and then there was a little peak behind it that they gradually came to realize their sera were also detecting, and that turned out to be a novel allergen from the PR5 family. So for, the protein in the center is actually a crystal structure of taumatin, but we've used that structure to do the modeling of JUNA3, which is taumatin, very similar to taumatin. Now, once we had established at the UTMB that this was an allergen and it was accepted by the IUIL, then people all over the world started identifying proteins that were very similar to it in very different forms. And of course, this is very confusing. If you develop an allergy, you have an allergy to cedar pollen, and then you can't eat apples or you can't eat green pepper. And you may think that they're separate allergies, but they could very well be due to the presence of these cross-reactive proteins. And actually, I don't know if maybe we can get the lights down, but just for speed, I will tell you that the Japanese patients and the Texas patients, even though they were separated by half the world, and actually I've given you a picture of the Japanese cedar and the Texas mountain cedar, and they're completely different species. They're about as different as you can get in the Pinaceae, or the cedar, and still these Japanese patients' sera had the same epitopes, so we could identify them on the dot spots that we did. So this indicated that indeed it wasn't just the overall protein, it was actually specific areas of these proteins that were allergenic. Now, I don't want to imply that there's no difference in the allergy sensitivity of Asians versus people in America. There's certainly a genetic factor in allergy as well. But in this case, we're able to show something at the 3D level of in the 3D structure of the protein, or at least in the linear structure. So then I'm going to spend the rest of the time talking about how the tools that have been developed in the Brown group can be used to break allergy into computationally solvable questions, naturally STAP, and then predicting allergenicity from the amino acid sequence, of course, the WHO rules and FASTA searches, but in more detail, how using the physicochemical properties of the amino acids rather than requiring simple identity can help you to identify common properties. And by the end of this talk, I do hope I've convinced you that we've identified at least one common property of nut proteins, or one common area of nut proteins that is probably responsible for cross-reactions, or some degree of cross-reaction. Now, I was switching to peanuts for the reason that peanuts are an amazing food source. And of course, if we could take the proteins out of peanuts, which are very stable proteins and incredibly nutritious, and put them into grains like cassava that have very little protein in them, we might really do an enormous service for areas of the world that will, especially with climate change and increasing desertification, if we don't do something about it, will become a great problem in the near future. So this little boy has been rescued by peanut paste, so-called plumpy nut. And this is just a wonderful thing to think that you can take these little packets and hand them out to the kids, and that they're going to survive a terrible famine and not die. I mean, this is great. I've always loved this picture. The only problem, of course, is that in Western worlds, and perhaps when we're feeding these children all this peanut, because they haven't been exposed to it perhaps when they were very young, we're going to have the same sort of anaphylactic things that are turning up within the United States and actually in the Western world. So this is the little boy showing typical signs of peanut allergy, but it's much more severe. This is an article actually from the 1960s in England. and where they actually were documenting 4 cases of people who died after exposure to peanuts. And of course now we have, this poor 7-year-old died recently just in this area, and she was in a school that had a peanut-free policy, so they still haven't figured out how she became exposed. So it's really a very serious problem. Now, using the STAP tools to try and explain, one of the big questions is why would somebody who's allergic to peanuts have an allergy at the same time to walnuts? Because maybe this little girl was exposed to a tree nut or some other nut that the parent might not have realized, and that she had a cross reaction. So it is important to know, or at least to sort of identify what are the potential cross reactions if somebody has a peanut allergy. And if you do just the fastest search in STAP, you can see that there are proteins in soybean and lentils that are closer to peanut than walnut. Actually, walnut's pretty low. If you will bear with me and just look at the BIT score and the E score on the far right there, what you will see is that the BIT score, even though these are big proteins, and we know that for Jugar-2, if you go down to English walnut, which is 12 on that table, the BIT score is only 100 for a protein of that size. And that's really remarkably low identity at that level. And also the E score is definitely great. But of course, that's based on the amino acids that will give you the secondary structure, which is the same, or the tertiary structure. And again, if you look at things like ARA-H2, if you want to predict cross-reactivity, and you know that the person's allergic to ARA-H2, if you put on some of these proteins, and this Jugar-2 N-terminal region, which is cleaved off in the mature protein, is one of the closest areas in walnut that match ARA-H2, but you can see there's an enormous difference. This is just an identity matrix. So they're only about 13% identical. So the question was, could we use then the PD scale and PCP to determine common properties that would be associated with these different allergens? And the way we were doing it is using the PD scale that Dr. Brown told you about, with the idea that, well, you know, if we, like in Dr. Seuss, you know, the evil things come together and they destroy the house. So if we knew thing 1, could we find thing 2, was the big question, and using the PD scale. So we knew that the IgE epitopes of peanut had been very, very well defined over the years, and actually Sohila was on a lot of the original papers defining those epitopes using the very tedious method of dot plotting. So knowing these epitopes, could we then use the PD scale to find things in nut allergens that might be similar in their properties? And now, just to show you, just because you have the same tertiary structure, it doesn't mean that you have the same amino acid sequence, even in areas that are fairly conserved. This is a ARA-H1 epitope. By the way, this is on our model of ARA-H1. And if you look at that structure, then you see that, and these are 3 models, all based on the same Viscellin structure. But what you see is, looking at the sequence, as you move from peanut to walnut is Jugar-2, and then into cashew, that area becomes more and more devolved. And according to our values, by the time you get to cashew, then it's over the PD value that we would say would be significant. So it's almost a PD value of 12. Whereas walnut's still in the same region. So now, this was our first validation, and this is published in Allergy in 2011. But what we did was start from an immunodominant epitope of ARA-H2, which is one of the major peanut proteins. And then we did the PD search to find similar areas. And we found that in Jugar-2, in this N-terminal region, if you look at the sequences here, you'll see that there's 1, 2, 3, and another one down here, 4 of these sequences with PD values up to about 9 that are similar to this. So they have low, so a PD value, the lower the PD, the more similar the sequence. But it was very interesting that they were all in this N-terminal region. So I decided, and then we were able to show that actually these sequences we could pick out with the PD value, so actually had cross-reactivity with patients, especially those who were allergic to both peanut and walnut. And this is in the paper with Suzanne and Sohila. And what was very, very interesting then was that we had indeed identified thing 2. So we were looking at epitopes in walnut that were cross-reacting with IgE. And furthermore, they were actually able to compete with ARA-H2 in the competitive ELISA assay to the same degree. And the numbers were pretty good. As Sohil has pointed out, there are 10 different epitopes known in ARA-H2, and we were able to compete out with the walnut peptide as well as with the peanut. So, but I went back then and I looked, at the same time I had been developing a method called physicochemical property consensus sequence for our virus database. So I have a database of flaviviruses like West Nile and dengue and all those other nasty polystrand RNA viruses. And the virologists had been saying to me, well, when you make that virus database, we're going to take all those sequences and we're going to look at it and we're going to determine what makes a virulent strain and a non-virulent. Well, the only problem was I only had 600. FlaviTrack now has something like 1,800 sequences in it. And if I wanted to look at those sequences working with Xerox to put it into microtype even, I would still need this wall to show those 600 sequences. There wasn't any way that any virologist in his right mind was going to be able to pick out point mutations in those sequences associated with virulence. So I realized that we needed some way for rational data reduction. And of course, I came back to this nice system of descriptors of the amino acids that Dr. Brown had, and we developed a new method for physicochemical property consensus sequence. Now, using this method that I won't go into, but it's described in several papers now, there's also a JMB paper on this. We took the Jugar2 NTP, so this N-terminal region that gets cleaved off. It's not part of the viscillin region of the protein, and realized that it was a trimeric repeat. And from that, we could align the sequences. I've taken the original sequences here and just boxed the repeat regions that are also present in other allergens to some extent. And then from these repeats, we could extract a PCP-consensus sequence that was closest in its properties to each of these 3 repeats. And then we took from the middle of that consensus sequence a small peptide and generated antibodies. So we made antibodies to a consensus. Now again, this consensus does not exist in nature. However, when you take it and you go into STAP and you use the PD scale, you find that there are sequences that are very similar to this sequence, so PD value less than 9. And I know now I'll have to break this down into more detail in terms of the PD value, just from the experimental data that I won't show you today. But basically what I was finding, especially the almond allergen Prudu-6 had 11 of these repeats that were similar to our consensus. And of course, in the walnut, there were several of them in the Jugar2 N-terminus. Buckwheat was interesting. And some of these repeats I could go back and show where it was known that they actually overlap IgE epitopes to some extent, certainly in the Jugar2. So we made antibodies in rabbit and eggs to this 13-mer. And then Sohila, what we're showing here is that these antibodies to this completely artificial sequence that has similarity to all these others indeed detected proteins of the right size in extracts of almond and cashew, not so well in peanut. One of the things is that I'm showing this as a structure. This is a model, and I took it out of FIRE for reasons I won't go into. But so FIRE is a website where you can just have something modeled if you want to. But the point I wanted to make is that this is probably either helical or unstructured. And then just from the middle of it, then we took this peptide to make the antibodies. So when I talk about now in the next slide, these are the antibodies I'm talking about. So we have now our antibodies to our artificial consensus sequence, and then this is a patient IG. The patient is allergic to almond. Are they also allergic to peanut? This patient, Suela, do you know? I should have asked you that before. Yeah, anyway.
If they're almond allergic, I don't know.
Okay, but I mean, the basic point is we have 4 almond allergic patients, Western blots from them that were actually done before we ever had the consensus sequence. But we could go back and show that the bands that our 13-mer was detecting were the same as what the patient was detecting in the Western blot. And then we've got mass spec data for a lot of these proteins now. We're getting ready to publish this data. Now, this is actually the crystal structure of the almond protein. And these purple regions here on the edges, these are the similar sequences. This is the crystallographic hexamer of the almond produce 6. And as you can see, they are surface exposed regions. And now I would like to say I'm showing you a structure, a biological structure similar to our consensus sequence. Unfortunately, every time I went into one of these crystal structures, exactly these areas were missing, which means that they must be disordered. They must be areas that the crystallographers are having difficulty defining the crystal coordinates for. And so, for example, with ARA-H1, we have a perfectly good crystal structure, but exactly those residues are missing, so I had to go back to our models to at least show you where on the structure these areas would be. And then this is a model of the walnut NTP for the same reason. So anyway, I would like just to finish by saying that I think that in the future, computational approaches of this sort, defining common areas of proteins, the things that look like each other and could be real markers for allergenicity, are going to be the basis for helping to consult with patients. And I think that the real use of the work that we're doing right now is in protein design, because we can identify allergens now. What's much less difficult to do is to say a protein's non-allergenic. And so what we would like to do, since we know that all of these nut proteins now have this common area, is to define exactly which sequences similar to this are very allergenic, so our patients are going to react to them all the time, and which may not be so allergenic, and therefore maybe just making point mutations in these regions could generate proteins that would be safer for human use and potentially prevent that little guy getting his plumpy nut from developing a reaction after he eats the package. Or eats the stuff in the package. Sometimes the kids eat the package too. So anyway, so just in conclusion, allergen IgE epitopes definitely have many common properties that we can identify at the level of the physicochemical property scale. And we can predict IgE epitopes. We've shown that, and also there's an earlier paper showing it for the cedar pollen epitopes that were identified by Terumi. And we can use the PCP-consensus method to define, once we have a set of motifs from allergenic proteins, we can use the PCP-consensus to define sequences that are most similar to them, and then moving out from that, determine how much distance you need in the physicochemical properties before a protein isn't allergenic anymore. So, and I'll just stop there, because of course eventually we'd like to go into the peanut breeding and walnut breeding and all these other things, and alter the proteins so they're not allergenic. And that's just another list of all our collaborators.